Hello friends, today the topic which we are going to deal is bacteriophage. So what do you mean by bacteriophage? Bacteriophage is a virus that infects bacteria and causes the lysis of bacterial cell. In other words, bacteriophage are viral parasites of bacteria. This means that bacteriophage just attacks the bacterial cell and utilizes its mechanism for its own growth. Who discovered bacteriophage? It was discovered by Frederick W. Swart in England in 1915. He observed some of the bacterial colonies which underwent lysis, and this lysis effect transmitted from colonies to colonies. Thus, he used some high dilutions of lysed colonies and passed them through bacterial filters. But to his surprise, it could transmit the lytic property. Therefore, he heated the filtrate to observe what about the lytic effect. But he observed that the lytic effect was lost. Thus, Swart suggested this lytic effect is due to virus. Significance of bacteriophages. Bacteriophages are nowadays used in viral research, genetic research, and viral pathology studies. Again, Frederick D. Herrle discovered bacteriophage in 1917 at Pasteur Institute in Paris, and he was the person who coined the word bacteriophage, which means bacteria eater. Characteristics of bacteriophage. Bacteriophages are widely distributed in nature and they can be easily isolated on laboratory. Just we need bacterial media in which we can grow bacteria and then use it for bacteriophage. They are made of nucleic acid core surrounded by a protein coat. They occur in different shapes and many of them have tails. This tail helps them in the inoculation of the host cell. They are of two types, lytic or virulent, temperate or avirulent. Lytic or virulent bacteriophage. When lytic phage infects cells, the cells respond by producing large number of new viruses. This is a pictorial representation of lytic cycle. The bacteriophage gets attached to bacterial cell. Once it is attached, it just inoculates its virus or its nucleic acid into the cell. Now inside the cell, a number of bacteriophages are formed. Then the cell lysis occurs and number of new lytic or bacteriophages are found, which again then attack the new cells and the cycle continues. Avirulent bacteriophage. Here the nucleic acid is carried and replicated in the host bacteria cell from one generation to another without any cell lysis. This is a pictorial representation of avirulent phase. Here you can see the cell is attacked by the bacteriophage, but the nucleic acid present in the bacteriophage just goes and get introduced into the DNA of the host cell, and the replication of the host cell continues and the bacteriophage DNA remains within the DNA of the bacterial cell. Then after certain generations, this avirulent phase can change into lytic cycle or they can continue to produce their DNA with viral phase DNA. Now we come towards the morphology of bacteriophage. The bacteriophage can be studied with the help of electron microscope. It contains a nucleic acid core that is covered by a protein coat, also called as capsid. Now the capsid is made up of subunits called capsomeres, and the capsomeres again contains a number of protein subunits or molecules called promoters. Their common morphology form have one head and a tail. Now here is the picture of a bacteriophage. On the top is the hexagonal head. Then comes the tail. The tail contains a contractile sheet, base pair and tail fibers. In this slide, we have shown all the 6 morphology. In our next slide, we will be explaining all the morphologies in detail. Here is the picture of 6 morphological types. The first one has a hexagonal head, a rigid tail with a contractile sheet and tail fibers. Now the second one has a hexagonal head, but it lacks the contractile sheet. Its tail is flexible and it doesn't have any tail fibers. And the third one is a hexagonal head and the tail shorter than the head. And the tail has no contractile sheet, but it may or may not have tail fibers. Now, the fourth one is made up of large capsomeres but has no tail. And the fifth one has a head made up of small capsomeres but has no tail. And the sixth one is filamentous. And last one is just a circle or circular in shape. Now we move towards the structure of phase. Phase have either cubic or helical symmetry. Cubic phase are regular solid or appear polyhedral, whereas helical phase are rod-shaped. Now in polyhedral phases are isohedral in shape with 20 facets, each of which is an equilateral triangle. These facets come together to form the 12 corners. In simple capsid, there is a capsiomer at each of 12 vertices. This capsiomer, which is surrounded by 5 other capsiomers called penton. In larger and complex capsids, the triangular facets are subdivided into a progressively large number of equilateral triangles. Helical symmetry. The rod-shaped viruses have their capsomeres arranged helically and not in staked ring. Example, Bacteriophage M13. Below you can see the image for the same. Some bacteriophage, example T-even, coliphages like T2, T4, etc. have complex structure, including a head and a tail. They have binal symmetry because each virion has both an isohedral head and a hollow helical tail. Nucleic acid of the phase. Tailed phases contain double-stranded DNA, whereas large capsomeres and filamentous one have single-stranded DNA. Some phases have single-stranded RNA, whereas DNAs of some phases are closed loop under certain conditions. Thank you, friend. Hope these slides have helped you for better understanding of bacteriophage. If you like our presentation, then please like, subscribe and share my channel. Also, we welcome your suggestions and comments. Thank you.